Hello everyone, welcome to SRIRAM's IS. We are discussing PYQ, basically the prelims through PYQ. This is an initiative and I'll be telling you about the important questions that was asked in the biotechnology section. So this is biotechnology part 3. We have already discussed part 1 and part 2. Hope this session that we are having, irrespective of all other subject, is making a purpose to you. Those of you who will be appearing for prelims this time, the purpose is to give you an insight how the question are being framed, what are the level of understanding that you need to have for each and every subject. Now, in align to that, biotechnology is a section which will help you to understand the level of question that is being framed, what you need to have an approach once you're reading the questions coming out from the biotechnology section. So, we'll start the session with some of the questions important, and this is the 3 session, biotechnology sessions 3, and I'll be looking into the questions that is from 2013 onwards. We have discussed the questions coming from 2014, right? So till 2014 can be accessed in the part 2 of the biotechnology. Now, talking about the other details specifically with the questions, it talks about the recombinant DNA technology, which is also called as genetic engineering, allows genes to be transferred. So this is basically a statement which is given, and you have to just analyze the statement which is given as per the question. So across the different species of the plant, it is asking, can the recombinant DNA technology allows genes to transfer across all different species of the plant? Yes, it allows, because the technology is such, it allows that the transfer can be done between the different species of a plant. Now the second is from animal to plant. Yes, from animal to plant can be done depending upon the requirement of the experiment that is taking place. So this statement 2 is also correct. Then we have statement 3, that from the microorganism to higher organism. Yes, this is also one of the significant feature of the recombinant DNA technology, or we call it as genetic engineering. So this has been an important topic, not only for the year 2014. UPSC has been asking questions from the technological part. The GNA slicing is there, GNA recombinant technology is there. So this has been frequently asked in the examination. So for this, the correct option would be D, that is 1, 2 and 3. I want you to read about the recombinant DNA technology, so that if any question re-emerged in the examination, you will be able to handle it easily. Now, this is the actual images to help you understand how the technology work in terms of recombinant DNA technology. You can see these are the bacterial cell, these are the plasmid which is actually there, and the insertion take place between this region with the DNA ligase, and further the DNA, the part of recombinant actions take place, right? So this is an image to give you a clarity on that particular aspect. Now we have second question, that is mycorrhizal bacteria technology or biotechnology has been used in rehabilitating degrading sites because of mycorrhiza enable the plants too. Now see, this is something very basics coming out of this particular question. It is asking you why this technology is being used, right? So this is resistance drought to increase the absorptive area, tolerate the extreme of pH, resistance disease of infections, right? Now this is again something important. If you look at each and every statement, this particular statement is correct because mycorrhizal technology use fungus, right? So it uses fungus, and this technology help in all these 3, tolerating the extreme pH, resisting the disease infections and resist drought. So for this particular statement, the correct option is D, that is 1, 2 and 3. Now, moving ahead with the other question, that is, what are the reasons for the people resistance? Now, just understand this question. This question is about basically to help you understand what led to the people to resist from the part of BT brinjals coming to India, right? So what are the reasons for the people resistance to the introductions of BT brinjal? Now this particular question was asked in 2012 because this was there in the news. Now it's time for BT cotton as well. UPSC has already asked BT cotton. The GM mustard crop which is there, this can be framed in the coming years, and specifically GM mustard become important, right? So in this particular statement is given that Bt brinjal has been created by insertion of gene from soil fungus in its genome. Now see, this is incorrect. If you look at the statement per se, the basic statement is incorrect because it is not from the soil fungus, it is actually the Bt, that is Bacillus thuringiensis. This is a bacteria which actually help it. So this statement is incorrect. Second, the seeds of the Bt brinjal are terminator seeds, and therefore the farmers have to buy the seeds before every season and the seed companies. No, this is not correct. This is absolutely incorrect because Bt brinjal seeds are not the terminator seeds, or it is also called as suicide seeds. Now third, what led to the people for the resistance of introductions of Bt brinjal in India? The third statement is that there is an apprehension that the consumption of Bt brinjal may have adverse impact on the health. Yes, this is true. This is something which are presumptions coming out from the people. I mean, people, if you're not aware, it's all about the general masses, that people are reluctant for the introductions of Bt brinjal as such. Now the fourth is there is some concern that the introductions of Bt brinjal may have adverse impact on the biodiversity. Now this is most likely to happen. That can lead to basically people being reluctant on that specific part. So as per this particular the statement number 3 and statement number 4 is correct. So the correct option here is 3 and 4. And if you use elimination technique also in these part, this can be easily negated. Moving ahead with the other question, this is from 2012 again. With reference to stem cells, frequently seen in the news, this frequently word is for this particular specific year. Which of the following statements are correct? Now, one thing also I wanted you to add on this because the question that was asked in the earlier years was lesser in number and they were very generic. Now, UPSC has made an advancement in terms of asking the questions, and the number of questions has also increased from 5 to 6, and it is taking a total of none less than 8 to 9, right? So make sure you are very aware of this all basic facts that UPSC has been asking the question on current affairs, specifically for the part of science and tech. Now we are looking into the stem cells. Stem cells can be derived from the mammal only. Now see, it is not only the mammal from which the stem cell can be derived. There are other various plants and animals where this can be done. Now stem cells can be used to screening a new drug. Yeah, this is true. It can be done for this specific purpose as well. The third is that stem cells can be used for medicinal therapies. Now what happens, like if you see abroad for US and country like Germany, they have been using the stem cell as a potential part where they have been using this in various other therapies as well. There have been various instances and evidences where this therapy of stem cell has been used. So for this particular question, the correct statement is 2 and 3, and we have option B, which is correct. Now, if you look at the other stem cells, I mean, how does it function? What are the cell that is considered as stem cell? Now, this can be specifically important for prelims, right? It can be asked that what are the stem cells? Now, brain is one of the stem cells as a part, neuron, right? Intestine, erythrocytes, hepatocytes, cardiac cell, osteocells, bones, and heart and liver. So make sure if a question is being framed about specifically for the stem cell, you are easily able to just answer it and understand what exactly is the stem cells. Now moving ahead, other than resistance to pest, what are the prospect of which the genetically engineering plant have been created? Now other than resistance. Now just understand this question. This was asked in 2012. You have to answer in a way that what led to this coming out from the genetic engineering plants. First is to enable them to withstand the drought. Yes, definitely this is one of the purpose. If you look at the part of resistance for a specific plant coming your way, it will ensure that the longevity and even the part of resistance is there with regards to drought. Second is to increase the nutritive value of the produce. Yes, definitely. There are certain parts where a modification is made to increase the nutritional aspect of a specific plant. The third is to enable them to grow and do photosynthesis in spaces and space stations. See, this is not the correct option because for photosynthesis you require light. Light is something which is inevitable and without light the things is not possible. So this statement is incorrect. The last statement is to increase their cell life. So if you negate the option number 3, this is something which is not there, and you have option left which is 1, 2 and 4. So option number C is the correct one. So in some question you can even use your common sense to come up with conclusions. Now moving ahead with the other question, are genetically engineered from the brinjal known as BT brinjal. You see, the question has been asked in BT brinjal, the part of BT cotton, the part of GM crops which is there. So UPSC has been asking question in civil services examination with regards to all of these crops. The genetically engineering from the BT brinjal, or the brinjal is also known as BT brinjal, has been developed with an objective of, what was the core objective? This was again asked in 2011. To make it pest resistance. Yes, to make it pest resistance is one of the reason and rationale behind coming to it. Second, to improve its taste and nutritive quality. Yes. Third, to make it drought resistance. Absolutely correct. And the fourth is to make its cell life longer. See, when it comes pest resistance and linking it to the cell life, these two can be a similar statement. So if you look at the precise answer, specifically for the correct answer is 1, to make it pest resistance, right? So this is the most appropriate answer for the BT brinjal and why the technology has been developed. Now, if you look at the functioning, the Bacillus thuringiensis, this is one where the virus, the bacteria is taken into consideration in the BT brinjal. And this is where the midgut or the insertion of monomer take place, and after all the part of solubilization and activations, the things take into this part and osmosis of the cell take place and further lead to the development of other BT crops. Now the other question, this is from 2011 again. At present, scientists can determine the arrangement of the relative positions of gene or DNA sequencing on a chromosome. How does it known to benefit us? Now first understand what this question is trying to test. This basic question is trying to understand the level of understanding of yours with regards to DNA sequencing. So it can be done. It is possible to know the pedigree of a livestock. Definitely it can be done because we have a diversity. Pedigree also help us to understand the various diversity coming their way. Second, it is possible to understand the cause of all human disease. Now, a very commonsensical question. See, all common and this type of only statement which is given will have to take a caution. We'll have to take a caution and understand on what context it is given. So, gene or DNA sequencing for all human disease, this is absolutely not correct. Now, third, it is possible to develop disease resistance animal breed. Now, this is correct. So, for this particular option, the correct option is 1 and 3, C is the correct one. Now we are coming to the other question from 2010. Genetically modified golden rice. Golden rice has been engineered to meet the human nutritional requirement. Which of the following statement best quantifies the golden rice? Now why and what was the purpose and rationale for the government to come up with the golden rice? First I will show you an image that will give you the clarity. You can see this is the golden rice. This is golden rice, right? And this is normal white rice. This is white rice, right? So this is you can see the images difference. Now let's look at the statement which is given. First, it is the grain have been fortified with the genes to provide 3 times higher grain yield as per the hectare and higher in the varieties. Now this has nothing to do when it comes to the nutritional aspect of any food. Fortified food, will come to fortified foods as well. Second is the grain contain provitamin A, which upon ingestion is converted into vitamin A in the human body. See, this is a concept. I mean, if you look at the part for fortified products which is there, government has come up with fortified foods. Now fortified foods are the one where intentionally or engineered way the nutritional aspect of the foods is actually being increased. So there are certain parts like vitamin, minerals, iron is added on the specific food. So that is a concept of fortification of the food. Now the third is it modified the genes cause synthesis to all the other 9 essential amino acids. Now this has nothing to do with this, and it's modified genes cause fortification of the grains with vitamin D. For this the correct option is B, and this is absolutely correct because in the golden rice the level of vitamin A was increased. Now when it comes to fortification, I want you to be very clear. There's certain food product that government has come up in the fortification. It might be fortified oil, it might be fortified pulses, right? And third would be fortified other food product, packet food products is also there, that the fortification is being done intentionally. In the midday meal scheme, government has also come up with some pulses and rice, which is iron rich fortified foods, to ensure that the children which is there at the midday meal scheme or availing this benefits are getting the adequate quantity of nutritional benefits. Now moving ahead with the other question. The other question is from 2007. Now you see, I'm trying to complete every question that is being asked in biotechnology section so that it is giving you a comprehensive part where you have been actually doing the series, so that it can help you and serve a purpose for the upcoming prelims examination. Now which of the following types used in the computed tomography employed for the visualization of internal structure of human body? Now tomography is something which is being used. The correct answer here is X-ray. I'll show you an image which will give you more clarity on that aspect. So this is how tomography is being used. See, this is a beam light which is actually being used, and X-ray is the ultimate source where the things are taken into consideration. This is a patient lying here, and we have motorized table and detectors to help it out. So the correct answer for this particular question is X-ray. Sound wave have nothing to do with this. Magnetic resonance is not the one, and radioisotope cannot be done with the patient's one, right? Now moving ahead with the other question, this is consider the following statement. Now this type of statement based question is still valid in the upcoming years. This was there in 2006, right? And most likely this will be the last session on the biotechnology. So just look at the questions that has been framed. So ELISA test is employed as a first and most basic test for the individual to detect cancer. See, ELISA test has nothing to do with cancer. It is basically used for HIV, AIDS and various other infections. The ELISA test is being used, right? So this is where you need to understand that this particular test is exactly not for cancer, right? This is incorrect. The second, almost 50% of human beings have Rh plus blood, while other have Rh plus negative, right? So it is asking that when it comes to Rh plus and Rh minus, which is rhesus factor, it is 50-50% that is being there in the statement, which is absolutely not correct. For Rs plus, we have, which is 85% of the people are actually Rs plus, no? And for Rh minus, we have only 15%. So in this particular case, both statement, neither one nor two, is correct, right? Now here we come to the other aspect of ELISA test. See, this might be important for the examination. There are 4 parts of ELISA test which is done. So this was the first statement that was asked. So I wanted you to be clear on this aspect as well. So there are 4 parts which is called direct ELISA. We have indirect ELISA, sandwich ELISA and competitive ELISA. You can check the images. You can either take the screenshot of the specific function. There might be a direct question asking you about the specific aspect of ELISA test. Which among the following is or are the ELISA test or one of the factors of the ELISA test, right? So here we come to the end of this session. I hope the entire session 1, 2 and 3 on the biotechnology has helped you. And this is, if you have not watched other sections and other parts of the biotechnology, do have a check. This will be something very comprehensive on your part, ensuring that you have actually completed all the biotechnology question that was actually being asked in the civil services examination conducted by the Union Public Service Commission. And it will also help you to get an idea, right, where we are lying, what are the practices that I need to have, what are the conceptual clarity that I need to think upon, and some basics will easily be able to answer. This is again a low hanging fruit. Ensure that you are doing well. I wish you all the best for the upcoming prelims examination. Thank you so much.